Forensic Lecture 7-3, The Role of the Toxicologist. Challenges facing the toxicologist uh, go beyond the analysis of alcohol. The toxicologist is confronted with a maze of drugs and poisons that could be present within body fluids or organs that is presented to them. They're required to examine the presence of any drugs or poisons that may be present within these fluids or tissues. The first process that they need to do is to do a general screening procedures to narrow down the thousands of possibilities that it could be to one specific drug or poison. They may only be presented with nanograms of blood to work with. And the other problem is there are a few substances that actually enter the body and completely leave the body as the same chemical. And so they need to have a thorough understanding of how the body alters or metabolizes the chemical structure. And this is essential for a good toxicologist. With collection and preservation of the evidence is really dependent on the attending physician, the medical examiner, the coroner, and the police investigators. It so the toxicology or toxicologist capabilities have to take into account all of these aspects, especially if we're looking at a living person or a deceased person. They have to be able to devise an analytical scheme to successfully detect, isolate, and identify the drug. Once the drug has been extracted from the biological fluids, then it can be identified. But there has to be enough blood presented to the toxicologist in order to perform these comprehensive analysis or the tests. Usually it's a minimum of 10 milliliters that's required. Techniques that the toxicologist may use is most of the time is through the extraction of the drugs that are based on them being acids or bases. So an acid is a component that donates hydrogen ions to another compound easily, and a base is a molecule capable of accepting the hydrogen ions that are shed by the acids. Chemists will then use a pH scale to measure the concentration of the hydrogen ions. Most of the drugs that are presented to the toxicology unit is alcohol, marijuana, and cocaine. They actually account for 90% or more of the drugs that they encounter, which can be used, which they can use the acid or base findings of it. And to do this, they control the pH of a water solution that the blood, the urine, or the tissue are dissolved in. Toxicologists can control the type of drug that's actually recovered. So acid drugs are easily extracted from acidified water solutions with organic solvents like chloroform, and the same or the opposite, basic drugs are easily removed from the basic water solutions with those organic solvents. Acid drugs are things like barbiturates, aspirin, basic drugs, PCP, methadone, amphetamines, cocaine, and so there's a wide number of drugs that they use the pH scale for to help extract from the samples. Strategies used for identifying abused drugs really entail two steps: a screening process and a confirmation, just like the other drug tests and alcohol. A screening test is employed again to provide the analysis with a quick insight into the likelihood of the specimen. Again, there are three common types: the thin layer chromatography, which we've already discussed, same as the gas chromatography. But then there's another one that's called immunoassay, and this is based on a specific drug antibody reaction within the blood. We're going to talk about that next chapter in more detail. But the primary advantage to this is the ability to detect small amounts of blood, or excuse me, of the drug in body fluids and organs. So it's much more sensitive. From there, any positive results are going to be considered tentative and have to be verified with a confirmation test. The confirmation test is, as we had discussed before, chromatography mass spectrometry, or the GCMS, and it's generally, again, accepted as a confirmation test of choice. So the gas chromatography separates the sample into its components, and then the mass spectrometer creates that quote-unquote fingerprint pattern used to identify the specific drugs. Drug testing is actually becoming very big in industries and governments as well, as for criminal matters. Urine tests are becoming quite common in job applications and employments, and both a screening and confirmation test have to be incorporated into the testing protocol to ensure the integrity of the test, especially if they're going to use any of that information against an individual if they are in violation. Another way that toxicologists can actually detect drugs is in hair. Most drugs remain in the bloodstream for only about 24 hours and only 72 hours in urine. So it may be needed to go further back in the history, and the only viable alternative is head hair. The hair is nourished from blood flow to the capillaries that are close to the root, and when there's drug present in the blood, it diffuses into the base of the hair. As that hair grows, it becomes permanently entrapped in the hardening proteins that are forming the hair. And so it becomes located on the shaft and becomes kind of like a historical marker, a timeline. The average hair typically grows about 1 centimeter per month. And so by analyzing the segments of the hair, it can give an actual timeline of drug use. However, the timeline can be distorted if drugs enter the hair from any environmental exposures or through sweat glands, but it is the only viable accepted process for drug use longer than 72 hours. It is not only the toxicologist's job to identify drugs, quote unquote drugs per se, or alcohol, although most of their time is devoted to that. They also have to verify other toxic substances. Heavy metals are one group of poisons that are encountered in murder cases. These include things like arsenic, mercury, thallium. Today, they're occasionally encountered, and this is because of the environmental regulations that are put on a number of our different substances used in our society, such as paints and so on. But to screen for these, the body fluid is dissolved in a hydrochloric solution, and a copper strip is then inserted into the solution. If the strip turns silver or has a dark coating, this indicates the presence of a heavy metal. And then, of course, a confirmation test is presented by analytical techniques. Most of the time, emission spectroscopy or X-ray diffraction. Another non-drug poison that they come into or encounter often is carbon monoxide. And carbon monoxide has the ability to combine with hemoglobin in the RBC. The RBC is a red blood cell, and the hemoglobin is what attracts the oxygen and carries it throughout the body. And when carbon dioxide forms or combines with the hemoglobin, it forms carboxyhemoglobin. Um, and what this does is it prevents the hemoglobin from combining with oxygen, and so the tissues basically suffocate. Asphyxia starts taking place, and there are two methods that are used to measure carbon monoxide concentration. The spectrophotometric method, which determines the amount of um, carboxyhemoglobin related relative to the oxyhemoglobin, so the hemoglobin of the oxygen combining with it. Or they can also look at the total free hemoglobins in there. The second method is gas chromatography, where a volume of blood is treated to release the carbon monoxide, which is then measured by gas chromatography. This is the levels of carbon monoxide are not clearly defined. Typically, a healthy middle-aged individual with a CO2 or a carbon monoxide blood saturation of 50 to 60% is fatal. If alcohol is involved, it drops from 35 to 45%. And interesting enough, chain smokers have a constant 8 to 10% of carbon monoxide in their blood from the cigarette. So it's not clearly defined, but it gives them a good starting point. We have or Law enforcement has drug recognition experts. In the 1970s, LAPD developed a clinical and psychophysical examinations to train police officers, um, and they will then use this to identify and differentiate between types of drug impairments. The program is now evolved into a national program to train police as drug recognition experts. It's about a 3 to 5 month training program. And once they finish it, they are considered a DRE, a drug recognition expert. The program incorporates standardized methods to determine whether individuals have taken one or more drugs. Each DRE has to complete a standard drug influence evaluation form, and it usually cannot determine which specific drugs are ingested, but it is reliable data from both the DRE and the forensic toxicologists um, that are required in order to prove drug intoxication. So it suggests it can suggest there's present of 7 broad categories of drugs: central nervous system depressants, stimulants, hallucinogens, dissociative anesthetics like PCP, inhalants, narcotic, and cannabis. Typically, the methods that are used are things like blood pressure, pulse, and pupil size to analyze whether one or more is taken in of these broad spectrums. But the components of the recognition process has 12 factors to it, and they must go through each of these 12 and complete the evaluation form as well. And then with that of the toxicologist, information from the toxicologist, then a definitive answer can be determined. That ends lecture 7-3. There's a quiz next class.